Okay, hello, and welcome to the script part of this week's module. We're going to we're going to talk about doing differential expression in practice. So starting out, there are a number of packages that you will need to install if you don't have them already. Now, typically speaking, you install packages once. And then when you're running your R script, you're going through it, you load that package into your environment by running this library function. The only time when you might want to install a package over again is if you want to update it to the latest version, because these package developers, they do release updates to their packages. And sometimes it's a small change, sometimes it's a big change, and you'll have to change all of your commands. So I try not to update my packages unless I really need to. But I have provided you with an installing packages R script. So packages that are from Bioconductor, which are these. They are going to be installed with this BioC Manager package. And if you don't already have that BioC Manager package, you would run this command to install BioC Manager from CRAN, CRAN. So run this first. We'll check to see if you have the latest BioC Manager. And then you can run one of these at a time to install these bioconductor packages. I tell you to run them one at a time because it's going to ask you, do you want to install or update these other packages that this package needs? And so you need to type something. If you want to install the dependencies, type something. If you want to update the ones you already have, there's options for that. It's going to ask you if you want to update the packages you already have. And if you just want to update all of them, type A. If you don't want to update any of them, type N. And you can pick and choose if you type S. But pay attention to those prompts, update your packages, install these one at a time. These last three, they are from CRAN, and so they have a different function to install them. So one way to check to see whether these packages have installed successfully after you run through that is to load them. So we load DEC2. It's saying that it's loading all of these other packages. If there was some kind of error saying, I don't know this package, or we can't load this package, that's when you might think that you haven't installed the package correctly. So watch as you're installing the package for errors. And then if you don't see any errors, try to load the package and check for errors then. So we're just gonna load all of these libraries. And so it's giving us some messages. None of these are errors. Here's a warning saying these packages were built under such and such version of R. Warnings are fine. I'm gonna load all of these packages. And so that's all fine for me because I know I already have these packages. But for you as a beginner, our user, you might not have the package. So we're going to close that. We're going to open this R script. And so this R script is what we're going to use to do the differential expression analysis for our lab. Okay, here it is. Okay, so you'll notice I only have the library functions. I only want to load in the packages that I know I have already installed. So as soon as you know you've installed the packages, you can close that. You can open up the R script. We're gonna have to load them again. I highlighted the whole chunk and I did Control Enter. So I'm gonna run everything I've highlighted. It's doing that same loading and warnings. It's fine. Okay, so we have our prompt here right now, so we know all of these loadings have finished. We are going to set our working directory right now. So I like to go through here, choose a directory. I know this is where I am already, but you might not be. You might need to move around to where you're actually working. Where are the input files? Where is your R code? Where are you working? You need to find that location. So here I've set my working directory. If I want to save that line of code, I would put it right here. So I'm going to copy and paste. There we go. Copy that, paste it in here. So I can run that again. It's already been done. That makes sure I'm in the right place. So the files to do differential expression, they're not huge, but they're a little bit larger than your computer might be able to handle. So as a help to you, the student, I've imported these this count table. I have done the conversion from, this is like log to counts plus 1. So we unlog the count with this, then we subtract 1 with this, and then we're going to round it so that there's no decimal places. The Xena data portal provides TCGA data as this log transformed count. And so we just want raw counts. And so we run this command to make sure the counts we are using are raw. And then I do a filter. I'm saying across the rows of my counts table, I want to add up all of the values in a row and keep the ones where that sum is greater than or equal to 2000. I save these index locations into this keep variable, and then I filter my raw counts table for these rows with these index locations. I save that into my raw counts table, and then I write the table to my directory. This is our filter counts text file. And this text file is what I have provided you on Carmen. So you are not going to run these commented out lines because I've already done it for you. If you were going to go to Xena and download the counts table straight from Xena, then you would need to run these lines of code. If you had a brand new counts table, you could go to code, comment, uncomment lines, and now they are live. You could run this chunk of code if you had the counts table straight from Xena. But since you don't, you would have to download that. You don't. You only have my filtered counts table. We're going to run this line. Run it. It still takes a little bit of time. It has a lot of genes. Okay, now it is. We see there are 28,000 observations. These are going to be genes, and there's 550 variables. These are going to be samples. What does that look like? Okay, so these are our genes. They're ensemble IDs. And these are our samples. They are TCGA sample names. Thing that I know is if the sample name has a 01 at the end, those are tumors. And if there is a 11 at the end, those are normal samples. The A here tells us which vial, which tube did this sample come from. Most of them are A's, but there are going to be some B's or C's scattered in there. Since there are so few of them, when we get to that point, I'm just going to keep the 01 A's and the 11 A's. But we can see how many there are of other types in here. We're going to import this gene annotation table. And this is going to convert ensemble IDs to gene symbols. Because gene symbols are a lot easier for us to see and kind of recognize. We also need a phenotype table. This is both of these are downloaded directly from Xena. Let's see, phenotype table. It has sample IDs, and then it has all of this clinical information. So this is a lung cancer data set. Do any of these samples have EGFR mutations? There's probably smoking. There's alcohol use. Yeah, number of pack years smoked. There's information about the tumor type, pathological T, path N. There's stage information. If you want to pan over, so this only shows the first 50 clinical phenotypes, you can pan over with this single arrow. If you click on the double arrow, it will take you all the way to the end. So this is the absolute last few columns of our phenotype table. We scroll all the way over. There's 125 columns in our phenotype table. Something you will notice, let's go back to the beginning. These sample IDs, they have dashes in them. If we look at our counts table has dots, and that's because when you import a table into R, it does not want dashes in the column names. It will convert any dash, any space into a dot. That's nice for column names, but it's hard if we want to try to link up these names in our counts table to these names in our sample table. Because I said in the lecture, for differential expression to work, you have a sample table and a counts table, and they need to have the same samples in both. And the samples need to be in the same order. So to do that, we need to match up the sample names. There's some lines of code here. So first, I'm going to do this string split. We're going to take the TCGA sample ID, split it by dashes, and keep the fourth part. What does this look like? This part. So we're keeping that very last part of the sample name. 01A, 11A. If we do table of that, we can see most of them are 01As and 11As. There's some B, D, E, other vials, but they're not very, there's not a really good sample size for any of the other vials. So there's going to come a line of code where we filter. We're also going to get the base part of the sample name. We're going to do the same string split where we split it based on these dashes, and we're keeping different parts of that TCGA name. And then we're building them back together. We're pasting them, and we're including the separator as a dot. Let's look at what that looks like. So we are breaking the TCGA sample name apart. We're building it back together with dots as delimiters, periods. Let's see. We're keeping these three parts. Yeah, so these base names should match our count sample names, and I think they do. They do. Okay, so that's good. We're going to take our phenotype table, and we're going, if we look right now, the row names are just numbers. We're going to take this new base column and set that to be our row names. Let's take that. We'll look back at it. There's our nice reformatted sample names. Here we're going to subset our phenotype table. We're going to use the subset function, and we want the part column, which we made up here, to equal 01A or part to equal 11A. And when you're subsetting like this, and you're saying, I want this column to equal such and such value, you need to use two equal signs. Always use two equal signs in any sort of logic statement. If you just use one equal sign, it's going to assign your column to be this value. We don't want that. We want a filter, so we're using two equal signs. Now our phenotype table, it went down in number of samples. If we run this table again, table for part, we'll see these extra low sample number groupings, they fell out because we took them out. And now we're left with 500 tumors and 253 normal samples. I did that here. We can see the same kind of thing if we look at a different column. This column has that primary tumor or solid tissue normal designation. We can see that. And then we can do a table with two parts. So we're saying, give me the count of records that have the different groups of sample type and the different groups of this part code. We run that and we see these assignments, they worked out great because all of the 01As are tumor, all of the 11As are normal. At this point, if we wanted to filter on something else, we could. That where, let's see, we already have that. On stage. Yeah. Oops, Mickey. No. Tumor stage diagnosis. Maybe we want this. What kind of tumor stage diagnosis groups do we have? Probably our biggest one is stage 1B. So what if we did that? What if we made all of our stages to be stage 1B? Stage IB. Or we want our tumor stage to be stage 1A. Both of those together. So we're going to lower our sample size just for this example of running. So we're lowering it down to 372. What's our table look like with that? So we have 240 tumors and 132 normals. And you'll see that I subsetted this phenotype table and I replaced it. I could have made a new table, but I don't want to go back. I'll never need to use that original phenotype table. So I'm safe to just replace it. If I wanted to filter this in a different way, like maybe I wanted to go back and add in stage just I couldn't do that. I would have to re-import because stage just I is not in here anymore. I'm going to have to re-import my phenotype table, do the part, do the base, do the row names, get this. I can run that, and then I can subset. I want this to be just I. Just I. Okay, so now we're including 4 more samples. That's what I'm going to run with. Okay, so we're at a point, we have filtered our phenotype table to some samples that we want to focus on. These are any sample with a stage I. So they're either IB, IA, or just I. We have made our sample names in a form that will match our counts names. They look pretty close. We're going to get a quick count of how many columns does our phenotype table have, because we're going to use that later. And we're going to make a variable where after we merge these tables, where will our genes start? It seems trivial right now, but it's useful information. Okay, so we've gotten our phenotype ready. Now we're going to make sure that our counts table has only the samples that are included in our phenotype table. There may be other ways of doing this, but what I like to do is merge my counts and my samples into one table to make sure they have the same samples and they're in the same order, and then I split them apart. So what I've done is I've taken my counts table and I've transposed it. What that will do is flip it on its side. So right now the samples are column names. When I transpose it, I'm going to flip on its side and the samples are going to be row names. Look at that. Yeah, it makes it harder to look at because there's so many columns. The columns are now genes and the rows are now samples. So the rows of our transpose counts tables are samples. The rows of our phenotype table are also samples. So what we're going to do is we're going to merge our phenotype and our transpose counts table by row names. If you remember, I told you when we merge, it makes this very annoying row names column. And we're going to push that back out to be row names by running this. So we can look at that. We have our nice row names now, and we're going to remove this column because we don't need it. So we're setting this funny row names to null because we don't need it anymore. So now that we've merged our two tables, we've reset the row names, we're using these variables of start of gene and total dimensions of our table to pull out our counts. So what we're doing, so our start of gene equals 128. If we look at our counts table, we're going to tab over so we can find that 128 column. I know it's a little bit silly. There's probably a better way to do this, but this is the way I've always done it. This is a 128th column. That's where the genes start. So that's where we need to start our counts table because we only want gene counts. And then we go all the way to the end of the table to this. We don't want that. We want -3 from that. So we want column 28262. Let's make sure we get that. Yeah. So we get the dimensions of our table. This tells us the number of rows, the number of columns. We get the second value of that. So that's the number of columns. And we subtract 3 because there's 3 extra columns at the very end of this table that just have to do with how the counts were done. I showed in the lecture, some reads are going to be no features, some reads are going to be ambiguous, and some reads are going to be multi-mapped. And this is telling us these are all the reads we couldn't assign to genes. So we need to throw those away. And so that's why we subtract 3 from the end. So we're pulling out only our counts. Similarly, we pull out only our samples. So from our counts and samples table, we need columns 1 to the number of phenotype columns that we have, which is 127. That's the very last clinical column. And after that, the counts start. So we're pulling out only sample information. All samples only. It's right here. We also have all counts only, which is here. I'm going to close some of this because I have a lot of tables open. Okay, so we've closed all of those. Now, the value that we're going to use for our differential expression analysis is this sample type. There's tumor and there's normal. It's not a factor right now. It's just a character string. If we were to factor it, just that column, and we're saving it back into our table. We look at our table now. It's going to set, it's alphabetical. So primary tumor is going to flip it around. We want to set the levels of our factor explicitly. So when we factor it, we say, I want the levels to be normal and then tumor. And this will make it so that a positive fold change is our tumor and a negative fold change is the normal. And the order that you put it in, I always have to look it up. So yeah, this will fix it. So we are assigning this. Let's look at our table again. And you'll see that instead of primary tumor and then solid tumor, now it's solid, normal, and then primary tumor. Been flipped around. And this is the order that we want it in. So after all of that, we have finished getting our tables ready. Now we can put it into our DSeq2 object. We need to transpose our counts table because right now this counts table has genes as columns and samples as rows. For DSeq2 to be happy, we need to flip that around. So we're flipping it back so that columns are samples and rows are genes. Our sample table is good because rows are samples and columns are clinical variables. And then we also tell it what is the column of our sample table that defines the groups. This should be a column that has only 2 groups and is available for every sample in your table. So we're going to run this. This is just building our object. It's going to give us a warning. So it's saying the groupings, the sample type dot samples, it has characters in these names that are not letters, numbers, underscores, or periods. And that's because we have these spaces in our group names. And it's just giving us a warning saying we see this, it's all right, but it's not a good practice. So if I were going to be doing this in my own experiment, I might replace the spaces with underscores and then it would be happier, but it's fine. It's all going to work. We're going to run these lines of code to normalize our counts, to estimate that variation in our data, and to do the statistical test. If we had wanted to, we could replace these 3 lines of code with just DESeq function. But I like to separate it out so we know exactly what we're doing. So it's going to take a little bit of time. Something that if you want, if this doesn't run and you want to make it faster, you would limit your sample size more. Maybe just limit it to stage IB. And then you would have a more focused sample set. These functions would run faster. But if you want to consider the whole data set, you're going to need a lot of computing power behind it, because it's testing all of these things. Something else that could lower the amount of data that you're using is to filter your counts table more. Maybe you want to increase this row sums filter to 5,000. You're testing fewer genes, your commands will run faster. So 2 ways to make your data set smaller is to filter your genes more heavily or to filter your samples more heavily. And I have had students try different TCGA data sets, and the breast cancer data set is so large that they have trouble running it on their own personal computers. Once we get into the Ohio supercomputer part of the class, you're going to have more computing power. There are options on the on-demand connection for the supercomputer where you can start a job of RStudio. It will run a particular R version. You can install your own packages. You can use data that's on OSC, and you can have a whole lot of computing power, as much memory as they have. And with that, you could run these commands a lot faster. Okay, so those 3 lines finished. This results also takes a little bit of time to run. This is what's going to pull the results information out of the dseek2 object. It's going to give you a table with fold changes, p-values, adjusted p-values, and it's all going to be based on the gene ID of your counts table. That's why it's important that we read in this gen code gene annotation table, because that's going to allow us to convert from ensemble IDs into gene symbols. And something that I've run into trouble with in the past is ensemble IDs are always unique, but gene symbols are not. So it's an example that I showed down here. I'm just talking to fill the time, but there are several listings, several ensemble IDs that all have the same metazoa SRP gene symbol. So if you end up with that in your results table, you're not going to be able to make your gene symbol be your row names, because row names in R have to be unique. So that's just an annoyance. And if you end up with some of those in your table, just a few of them, you might want to take them out, because this isn't even a protein coding gene that we're really interested in. So be aware that gene symbols are not always unique. And if you have multiple genes with the same gene symbol, you wouldn't be able to set them as row names. Okay, so where do we go? We have res, which is a dseek results object. It's going to have all this information about fold change, but it's still not very useful. We could get a summary of res, which I showed in the lecture, but the way to pull out the information to be useful is we're going to convert it to a data frame right here. And now we have a table called result, has 6 columns, which has the average of this gene across all of our samples. It has the log2 fold change, and it has the p-value and the adjusted p-value. So this adjusted p-value is what we're most interested in. Also the fold change, but it has ensemble ID names. So we're going to merge our result with our gene annotation by row names. We're going to assign this new column of row names to be our row names. Oops, I can't do that. Okay. And then we're going to remove that column. So if we look at our results plus genes, it has more information now. So it has these columns from our gene annotation, and it has these columns from our result. So that's a really good table. We're going to write that to a text file now. You run this, it saves it to your working directory. Now, the first visualization we want to do is to plot a PCA. And to do that, we need to pull the expression table out of our DEC2 object. We're going to apply a new normalization method to that object. It's called variance stabilizing transformation. Here's the information about it, but it's basically normalizing your counts and doing a log2 transform. We do that, we apply it to our object, and then we pull out the expression with this assay function. There is another similar type of transformation provided by DSeq2. It's called rlog. And people say it's stronger, but it takes a lot more time to run. So VST is a fine alternative. So now we have MAT. MAT, let's close this res. We have MAT, which is an expression matrix. Has columns of our sample names and rows of our genes, and this value is normalized expression. And this is what we can use for plotting our PCA plot. I'm going to leave that up to you to plot and to make some inferences, some commentary on what you see. What do you think happened? We're shading it based on samples. What can you conclude about the PCA plot? Similarly, I set up a volcano plot for you using that table that has the gene symbols, our fold change, and our adjusted p-value. We specify the columns that we want to use for fold change and p-value. And then this is an optional part. We're going to ask it, label these gene dots based on the gene column. And if you remember from the lecture, there are a lot of gene symbols in the volcano plot. And including this option, it's going to label as many dots from those significant quadrants as it can. The ones with the smallest adjusted p-value and largest fold change. If you didn't want to label the genes, we could do this. We're going to take out label, but we need to include a placeholder. So this is going to make that volcano plot with no labeled genes. Okay, so I'm not plotting them, but I want you as the user to plot them to make some observations about what you're seeing. Okay, so the third plot we're going to consider is a heat map. And I've told you we need to get a subset of genes for our heat map. So we're going to filter based on the absolute value of our fold change and the adjusted p-value. First off, I'm setting this fold change filter to 0. So it's not going to filter anything at all. It's just going to keep everything. And the adjusted p-value to be 0.05. So this is the absolute lowest filter that I would even consider. We save that as significant results, and we ask, what are the dimensions of this significant results table? What are the number of rows? So this is going to tell us the number of genes that meet our criteria. And it's ridiculous. It's 18,000 genes. It's going to take forever to plot a heat map with 18,000 genes. So we're going to do something more strict. My example is FDR 1 times 10 to the negative 50 and fold change of 3. If I filter based on those, so here we have variables for fold change and FDR, which we've set up here. We run that. This is a much more reasonable number of genes. So 364. I think that's great. 364. Where is that table? Significant results. Yeah. These are most differentially expressed genes. I'm going to leave it up to you, the user, to select cutoffs that you want to visualize. Set some cutoffs. Make sure it's less than 2,000, because more than 2,000, the program will complain. So set some cutoffs, see how many genes you get out of it. We're going to get a variable of number of samples. Number of samples, 267. We're going to merge our significant results with our expression matrix. So we have our significant expression table here now. Has the row names. We're gonna fix that. Didn't we even do this? Okay, so we can do that. I have set the row names of our significant expression table to be gene symbol. This is optional. You don't really have to do this, but if you want to include the gene name in your heat map, maybe you only have a few genes that you're plotting, you can leave that gene, that row name in your heat map. When we have 300 or so of them, it's not going to be feasible to keep all of those in our table. So when I scale it, I'm going to remove those row names. When I ran this before, there were some of those genes that have multiple records, so I just removed them. I don't think that's an issue here. I don't think they're even in my table. We can still run it. There, I set the row names to be our genes, which I've already done. We also need to clean this up a little bit. We remove this column, is right here. It's the ensemble IDs. We're going to remove that. And we're also going to make sure the significant expression table only has these numbers in it. If we go back to the end of our table, there's some information here that is not normalized expression. So when we do this subset of our table, we're selecting only the first few columns that we know have normalized expression values in it. So we limit that. We can look back over here, and all of that is gone now. So we know that our entire table is only normalized expression. Now we're going to scale this table. And I do a little bit of transposing to make sure it scales properly. So right now it has columns as samples and rows as genes. We flip that on the side to do the scale. We want samples as rows before we scale. And then after we scale, we're flipping it back up. Hopefully that makes sense. We're doing this all together in one line. And this just makes the gene so that the average of the gene is centered around 0. Above average for that gene is positive, and below average for that gene is negative. If you do this scaling and you plot your heat map and it just looks really strange, then you might have scaled in the wrong direction. Now, this next line of code, I build an annotation track for my samples. The nice thing about this package that I'm using for heat maps is called complex heat maps. Yeah, complex heat map. It has a whole variety of different annotations, setups that you can make, different options, different palettes for coloring, and all of these different things. You can cluster your rows, you can cluster your columns. You don't have to. So if you're interested in building different types of complex heat maps, go to that website where they have all of the options. Find the option that looks most like what you want to accomplish. But we save this annotation track. It's going to set it up using our sample type samples, and it's going to say, I want to set up solid tissue normal to be gray. I want to set up primary tumor to be dark green. Change this to different colors. I want it to be dark blue. Whatever makes sense for your, whatever you're trying to do. And then we run this heat map command. We give it our scaled expression table. We give it our annotation for our samples. We're going to tell it, I don't want to see sample names. I don't want to see gene names because there's 300 or so of them. That's just going to be way too crowded on our rows. Rows are going to be genes. I got it flipped. Okay, so no gene names, no sample names, just because it's too crowded. I'm going to have the name of my shading legend to be VST normalization norm, because that's what we're plotting. It's actually scaled VST norm. And then we have a title comparing tumor versus normal. You might want to add fold change of 3 and FDR of 1E negative 50, whatever your filter is. And I include this \n as a new line character. So our title would print this, would make a new line, and then it would print the other side. But that's optional, you could add. And that's a heat map. I'm going to let you plot your own heat map with whatever your filter cutoffs you want and make some commentary about your heat map. To make some box plots, I merge the sample table with the significant expression table. This gets our normalized expression together with the sample information. And this is that one-liner that I've talked about before that you can do to merge tables. It will merge your tables, set the row names to be this capital R row names, and then set the capital R row names to null to remove that silly column. And so it does it all in one line. But for me to look up this one line or to type out the 3 lines, it's easier for me to type out the 3 lines. But this is an example. This puts all the data together so we can plot a box plot. If we look at the results, I think the actual, let's sort it based on fold change. The actual most negative gene and most positive gene, they're a little bit different. So if you wanted to check out your results, plot the most positive and the most negative, you could. This is still using my old genes. You would change this TICRR to be whatever your gene name is. You could also change it in the title of the plot and make it make more sense. We have map. No, it's probably not there. TICRR. We plot this one. It looks a lot like what I had before. Very positive, very high expression in the tumor group, lower expression in the normal. Tumor name got cut off, but if you click on zoom here, you can see them both there. Same thing with this other gene that is it's not even in there. So let's get another gene to put in here instead. A negative gene. Let's see. PRG4. I can remember that one. PRG4. We're also going to change it in our title just so it makes sense. PRG4, it has high expression in our normal, zoom, and lower expression in our tumor. So this is a negative fold change, where the other was a positive fold change. Okay, so we've done the differential expression, some visualization, and then the last few chunks of code. So this is pulling back in the file we did earlier. Maybe you didn't have time to do all of this in one setting. You did up to exporting the text file somewhere up here. Yeah, we wrote this table. maybe tomorrow you came back and you didn't want to run through the differential expression again. You could pick back up, read the table in. You still have to load your libraries. Load your libraries, set your working directory. Then you could pick back up with the table that you made the other day. We're going to import this. Now we have the table saved as DEG results. You're going to filter these results with however you want to. Try to aim for between 200 and 2,000 genes. With this, I get 364. And then you input your gene list. You tell it biological process or cellular component or molecular function is what I already have in there. You run that. Going through, there's 364 genes. If your gene list is too small, you might not get any results from the enrichment. Perhaps nothing was found. Let's check out, we're saving this into an enrich result object called ego. You check out, yeah, we have results. Okay, it's always microtubule binding. It's not a great way to look at it, but we see we have results. We can simplify our ego to collapse any of these terms. So maybe microtubule binding and tubulin binding get collapsed together. They probably have the same 23 genes. Let's see what that happens, how that works. Okay, let's head simple ego. What does that look like? True? Yes, so tubulin binding is gone now, and we're just left with microtubule binding. So we could print out the first 10 lines of the simple ego to my enrichment text file. We do that. We can plot a bar plot of ego. We can plot a bar plot of simple ego. We could also do dot plots. Dot plot. So the x-axis is the gene ratio. The y-axis are these different terms. The shading color is the adjusted p-value of the enrichment. I also have code for splitting this data into positive fold change, upregulated, and negative fold change, which I call downregulated. We have a new table. So these are our 364 genes that met my criteria. I have a new column name called group, which I've assigned them each as upregulated if the fold change is positive, or downregulated if the fold change is negative. And then I do a compare cluster to relate this group of upregulated or downregulated to symbol. And that's what I show in the lecture, showing gene terms that are enriched in our negative fold change group and gene terms that are regulated in our positive fold change group. So you can play around with that. Sometimes when you plot these plots with cluster profiler results, the names of these terms, they get all squished up. They overlap some. If you want to print the name of this term all in one line, you'd install this stringer package. I think we already loaded it up above, and you add on this bit of code. All of these plots are based in ggplots too, and so you can add things to them. And we are telling R, I want you to make this y-axis label to have a wider width. So doing this would stretch out the term name. Can I run this? I don't think I have. Let's do simple ego here instead. There we go. So it's going to give us a bit of a warning. It's fine. The warning's fine. You'll see over here, we see our term names, but all of the data is squished up. If you click on zoom, there, now we see it. It's a lot nicer. All of these are one single line, and all of this information over here, it looks really nice. So if you were to print this to a file, you could make the height of your picture be shorter because there's these names, they're not taking up multiple lines of space. So you could think about, what does the size of my plot need to be? And if you want to stretch out your gene terms, it makes it look a little bit nicer. Okay, so this is running through the script of how to do differential expression analysis. Thank you.